Good morning. It's 9 a.m. here in San Diego, and welcome to today's webinar. Today's webinar is on flexible induced fit ligand docking and screening. Just a quick slide about MolSoft. The company was founded by Ruben Abagayan, who is a professor at UCSD. Our principal scientist here is Max Totroff, and our principal developer is Eugene Rausch. And these three are primarily responsible for the development of the software. MolSoft is located in San Diego, in California, and our software is called ICM because it's based on the internal coordinate mechanics. My name is Andrew Ori, so if you have any questions after the webinar, please feel free to email or call, and we will progress. So if you have any questions during the webinar, you can use the question panel, or you can raise your hand at the end and I can open your mic. Everyone who joins the webinar is free to download the software and use it for 1 month, 1 month key. You just need to go to the support page, download ICM Pro, which is the full package, and then email me the host ID that is reported. It runs on Windows, Mac and Linux. So this webinar is the third in the series. We had one on the 3D ligand editor, which allows you to optimize ligands in the CIDR binding pocket. And last week's one was on docking and screening. So they're kind of prerequisites for this one. But if you want to watch those again, you can go to the YouTube and just search MolSoft and you'll find the previous webinars. So today's webinar topics. We have going to start with the introduction to some of the methods that we have incorporated in ICM. We are going to look at some different methods for incorporating flexibility during docking. So one of them is called 4D docking, which is basically a multiple receptor docking. So you could be in a scenario where you have one or more crystal structures of your receptor that represents the flexibility of the binding pocket, and you can dock to those automatically in one go. Or alternatively, we have explicit group docking, where you can allow certain selected residues, selected groups like hydroxyls, for example, to be fully flexible, or water molecules to be fully flexible during the docking. These two approaches are also included in the ICM 3D ligand editor, which we developed with medicinal chemists at Novartis. And so you can use the flexible docking to optimize leads, which we'll show. And there's another method that will open up your pocket and refine it. If your pocket is particularly closed, for example, it's called SCARE, and it's called it's a dual alanine scanning and refining method. And then we'll talk a little bit at the end about how to generate multiple receptor conformations if you're not in the position whereby you have crystal structures that represent the flexibility. There are some ways in ICM that you can use to model the flexibility. So if you're not familiar with induced fit, the original model was Fisher's lock and key, whereby the ligand would bind, match perfectly, but in reality that's not the case. Usually binding is more related to Koshland's theory of induced fit. So when the ligand binds, the receptor changes shape to accommodate the ligand. So it's this kind of docking that we will be doing today. So we're going to start with receptor ensemble docking. So we have two different approaches which you could use in ICM. One is 4D docking, which is a single run, and one is multiple receptor conformations. So you could imagine if you're in this scenario where you have 3 crystal structures that represent flexibility within the pocket. So maybe there's a loop region that comes in and out in each one, or there's a flexible receptor. So what you could do, if you wanted to, what we used to do in fact, was dock independently to each of these 3 conformations. So it's a bit time consuming. Then you have to merge the results and process them. But what 4D docking does is allow you to screen to all 3 of these structures in one go, in one run. and it's actually more efficient. The convergence time for the 4D is a lot shorter, and you get the same similar results as if you're doing it individually. And this method, 4D docking, was published by Giovanni Bottagoni and Max Totroff and Ruben Abagayan. So the receptor ensembles, 4D grids. Four-dimensional docking is a fast and accurate account of discrete receptor flexibility in ligand docking. So to do that, we place all the receptors that you want to use to dock to into one single, what we call stack. So you have you have one object similar to like a PDB, but that one object would have multiple conformations of your receptor within it. You can also have conformations of your receptor with a water molecule in and out. That could be one of your ensembles. And then ICM calculates the docking energy grid maps for each of these pockets in one go. But then they're stored as a single grid that you can dock. So this is kind of the fourth dimension, why we call it 4D. And then during, if you're present at the webinars, or you can look at the YouTube videos, but we use a Monte Carlo bias probability method developed by Ruben and Max back in '94. So during the Monte Carlo simulation, there's an additional step, a stochastic step, which jumps between the different 4D layers or the different receptor conformations during docking. This is benchmarked in this paper. 99 therapeutically relevant proteins were used, and 300 diverse ligands, and 77% complexes correctly reproduced. And on average, fourfold faster than doing the independent grid docking. So today's example in ICM, we're going to look at the estrogen-related receptor, gamma. It's quite a challenging example, so I'll show you why. Okay. So we go to ICM, and this example is actually online if you want to look at it as well. I can send you the link to download it. But basically, to set this up, all I've done is read in 3 estrogen-related receptor structures, 1 S9Q, 2 GPP, and 2 EWP. So I went to search and read them in. I then actually converted them to an ICM object, something we covered in the previous webinars. So to do that, you just right-click here and say convert. This will add hydrogens and optimize some side chains. For this example, I'm just going to delete actually some some chains we don't need. I'm just going to quickly delete. So select them, delete. Okay. So then we're left with 3 structures here. So the estrogen-related receptor is actually an orphan receptor that acts as a constitutive activator of transcription. There's no endogenous ligand known, but is reported to interact with several modulators, which we can see bound to the binding pocket here. It's interesting. It's an interesting example for this webinar because there are actually 2 distinct conformations, or between agonist bound, whereby you can see this helix here is inside the pocket, but when an inverse agonist binds, this helix flips out, so you can see the blue one sticking out here and the pink one here. So with the inverse agonist bound, they shift. So it's quite an extreme case of receptor flexibility. So we can also look at the binding pockets for each of these conformations. I'm just going to show the agonist bound, which is 2 GPP. We go to Tools, 3D, Predict, ICM Pocket Finder, and go OK. We should see the binding pocket here for this one. And for the inverse agonist structure, 19SQ or the other one, we can just select, go to Tools, 3D Predict ICM Pocket Finder, and run ICM Pocket Finder again. And then we can see the differences in the pocket. If I just color this, if you right click on the pocket, you can change the color. And you can also change the style of the display. So if we compare the blue one, which is the agonist binding pocket, with the red one, you can see there's a dramatic difference. In actual fact, the main, obviously that helix is moving out, but one of the side chains, phenylalanine 435, if we look at 435, you expand here, you can find the, I'm looking for F435 here. If I display that in stick, you can see it here. And if we compare it with where the position of this is in the agonist structure, so this phenylalanine moves in and out, which shifts that helix out of the pocket. Okay, so that's the main reason. Okay, so how would, if we're imagining this is our problem, our docking scenario, we have a flexible pockets. How would we dock efficiently to these three structures with different pockets? Okay, so what we need to do first is to make sure that the structures are empty. There's no ligands in them. So I'm just going to, for clarity, I'm just going to undisplay some things. So I'm just going to, each of these structures is bound with one of the ERR modulators. So I'm just going to right click on the ligand and say move from object. That moves it to a separate object. So now this is empty, this structure. And same with this one. Move from object. So now I have the ligand here. This is empty. And the same with the third one. Move from object. So now we have the three ligands here, which I can actually undisplay because we don't need those at the moment. And then we're just left with the crystal structures here. So in this example, we've actually pre-superimposed these structures. But for 4D docking, you need to make sure that the superposition is reasonable, particularly around the binding pocket. So to do a superposition, you can right-click and drag, or you can make a selection, and then go to Display, and then Superimpose. Click on this Superimpose button here, and it asks you which structure you want to be static, and the others will be superimposed on it. It's quite a useful tool. So it basically superimposes anything you select. So if I want to superimpose this loop region, just select the loop by right-clicking and dragging, click Superimpose, and it will superimpose that region. But for this example, we need the binding pocket to be superimposed. So I'm going to select here and choose Superimpose. Okay. So as I mentioned, the way the 4D docking works is that we place all these structures into one single object. So then ICM can dock automatically to this one single object, and we call that a stack. So we need to build that stack. So we look at the three structures we have. We have 2EWP, which has 224 amino acids in it. Has a 2GPP is a chain of 225, and 1S9Q is 224. So usually it's best to choose. We have one. You have to choose one of them as being the main object. So we'll choose the one with the longest chain, which happens to be the agonist structure 2GPP. So that means that we'll place this structure into this one, and 2EWP into 2GPP. So to do that, we need to make some selections. So I'm going to select the two structures. The first one, the two inverse agonist bound structures, 1SQ9 and 2EWP. You can do that by clicking and then press Control, and you can make a non-contiguous selection. And then I'm going to place it into what we call an orange selection. It's just a way of making a separate selection. So so you can see at the moment this is green, and then there's this button here that says green to orange. If you click on that, it will make all those selections orange. Okay. And then I need to select the 2-GPP, the agonist bound structure, just by double clicking. So now we have green selection and orange selection. We go to the mole mechanics menu and choose impose conformations. It's asking us which would be the main object. So we said that the main object would be the one with the longest chain, which is 2-GPP, which is a green selection. You see the green cross here. And then the source objects, which are going to be placed into the main object, are the orange selection. So if you use the drop-down button here and choose the orange cross, you can see two selections. So then you can superimpose, but we've already pre-done that, so we don't need to do that. And you can also re-optimize the hydrogens. Most likely you've already optimized them. There's no harm in doing that, it just takes a little bit longer. So I'm going to uncheck it for the case of this example. So you go OK. Right, so now you can see now in our main object, that's going to get rid of all the selections. Now you can see in our main object, the 2-GPP, we have the chain, the A chain, we have the two pockets that we found, but then we have an additional thing here called the stack. So in the stack we now have the structure for 1S9Q, which is the inverse agonist, and the other inverse agonist structure 2EWP here. So we can toggle through these by clicking, you expand on the stack region here, and then you can toggle through each one. It's going to undisplay the other ones. So this is 2-GPP, the original structure. Toggle through to the next one and next one. So ICM will dock to all three of these structures. I had a question about the maximum number of structures that you can use. You can use as many as you want, basically, but it's usually best to choose a small subset that represents the flexibility within the pocket. So probably no more than 10 would be, you can use as many as you want, but 10 would be a reasonable cutoff. And hopefully those would be diverse structures, so you'd have one with the loop in or one with the flexible residue in and out, and what have you. But if you had 10, obviously you'd read 10 in, 10 structures in. I had a question, some other questions. Yes, so the stack is just a question about the stack, and the stack is just the PDB structures all in one file, basically. So if you had a pre-calculated stack, then you could just read it in, and you wouldn't have to go through this step. You can also play the stack, and that will show you the flexibility. You can see the helix 12 moving in and out of the pocket as well. If you wanted to record that to a video, there's video options down the bottom. That's another story. So this structure now is ready for docking. So last week we went through the docking menu. So we go to docking, the docking menu, and we choose new project, and we need to give the docking project a name. I'm just going to call it Webinar 4D. And then it asks us for a project folder. So where are you going to store all your docking files? Just my project folder. You can change that wherever you want to place it. Then it's asking you which is the receptor object. So this is critical. So the receptor object, which has all four, which has a stack in it, is A_2GPP. And it's asking you where the docking site is. So we know, so the docking site, we can use this pocket to define the docking site. So the pocket is G_pocket2GPP, and we need to use the drop-down button here, and it says define site around a mesh. We call these meshes, And you go, okay, so now you can see we have a selection around here. Okay, so now it's ready for docking, but you need to uncheck this option called Make Receptor Maps immediately. You can do that, and it will just make the receptor map for the first object in your stack. But because we're going to do 4D docking, we need to remake those maps for each one. So I'm going to uncheck it for the first step. Just go OK. So as last week, we have this probe where you can use your middle mouse button to move it. That's where the docking will start. It's generally fine just to leave it where it is by default. And then we have the purple box. So if you have a very flexible structure, you need to make sure the purple box covers the binding pockets for each of the structures that you have. Probably if you choose the largest pocket out of the 3 structures, then it will be fine. But if not, you need to change the distance. Okay. Oh, yeah, I had some interesting questions. I'll get back to them probably at the end. Yeah, I'll get back to some of these at the end. So so now it's ready for docking. Now we need to tell ICM that we want to do 4D docking, not just regular docking to one structure. So we go to docking and we go to flexible receptor and we say set up 4D grid. And we want to make the maps for the 4D. So just go OK. And that would generate this will generate maps for each of the 3 receptors around that binding pocket which we selected. You had one question about molecular dynamics. So if you have representative structures from MD, yeah, you could just read them in to ICM. If you can read them in PDB format, and just basically do exactly the same as what we've done so far. That's not a problem. You had a question about maximum number of conformers. So 25. I'll show you the key thing. If you've got a large number of conformers, you need to make sure the docking simulation is for a reasonable amount of time. I'll show that in a minute. Okay, so you can see now that we've set this up, the docking is set up. So now we have a new object in our ICM workspace. It's called Webinar 4D because that's the name of the docking project. It has our stack of 3 conformations in it. So we know it's all set up. And so basically we're ready to dock. I'm just going to, so we have these 3 modulators that we took out of the structure. I'm just going to extract them to a chemical spreadsheet to dock them, redock them. So you can select them, and then you right-click and you choose Extract Ligand as 2D drawing, and now we have a chemical spreadsheet here with those 3 modulators. So one of them is the one from GPP. This is an agonist. This is a ERR agonist. These are inverse. The first one and the third one are inverse agonists. So we can dock this table to our 4D structures. So to do that, we go to docking and we choose I'm going to dock a chemical table. But you could just choose from an SGF file that you have stored on your computer, and you can dock from SGF files. But I just read in a table, so I'm going to go dock chemical table, and there's a tab called current project, which is what we're going to dock to. The table we're going to dock full of structures is called chem, so that's fine. I was going to uncheck the option dock immediately, because if I press it then it's just going to use the default parameters for docking, but for 4D docking we don't want to do that, so we just go OK. And then we get this little table at the side of our docking table that we're going to dock. Now the thoroughness, this is the length of the simulation. Usually for virtual screening to a single structure, 1, 2 or 3 is a reasonable number. But for 4D docking, because you have the fourth dimension, which is like the multiple receptor conformations, you need to increase the length of simulation in order to find the correct binding pose. So for 3 structures, something like 10 would be a reasonable number. You can play around with the thoroughness. If you have a complex with a known modulator, in this case we have a modulator bound, so we could play around with the thoroughness to see, if we need to increase it to get a decent redox ligand. So then we go, basically you just press dock and the docking will run. So if you have 25 conformations from NMR, then you would definitely need to increase this to a higher amount. In fact, probably from the NMR, you'd maybe cluster the structures and then just choose maybe up to 10 representative structures within the ensemble. It's kind of a subjective thing to do. We do have a method which we'll talk about actually in the seminar webinar series after the summer. We're going to do a lot of modeling, and one of those things is how to there's an option here called select subset of pockets. So it will select a diverse range of pockets for docking, basically. You can use that. You can play around with this, but that's not part of this webinar. Okay, so the docking is ready. You press dock, and then you get the results. And that takes a little bit longer to run because we are using a thoroughness of 10, and we're docking 3 ligands. So I have the results here. So when the docking is finished, you would go to docking and choose make hit list. That's all I've done here, basically. Or if you're docking from the table, the hit list is automatically generated. And from last week, you may remember that the lower the score, the better the predicted interaction. So you can sort. This is our hit list of docked ligands. So I was going to undisplay some stuff here. It's going to just leave the receptor object. This is the this is the docked object, because you can see in here we have the 3 stacked conformations that we can toggle through if you wanted to. So our best hit, it came from PDB 2EWP. So hopefully if the docking simulation worked correctly, then it will find that ligand in the correct receptor conformation, which is 2EWP. And you can see that we have the energies here for hydrogen bonding, van der Waals interactions, electrostatics. We went through these last week. At the end, we have another column called receptor conformation. And you can see in here we have number 3. So number 3 represents the 3rd member of the stack. And we can see, probably not very well in via GoToMeeting, but here we have the stack, and each stack is labeled. Number 3 is 2EWP. So if we click on this ligand and load it, we should see the stack jump to number 3, which is 2EWP, and you can see the ligand bound. Same for the next one. This ligand is this is the agonist, the ERR agonist from 2GPP. So if you click on here, it's actually RecConf1, and it will jump to RecConf1 here, so you can see it prefers 2GPP. Okay, so you have the score, and this is your hit list. So that's basically how you'd run it. You can run, as I mentioned, you can run from batch. You can do it virtual screening, large data sets, to multiple conformation receptors. And this approach has been very successful in the D3R blind docking and screening competitions, whereby pharmaceutical companies provide complexes which aren't published, and we compare our method against other methods. And ICM has been performing very well, coming in first place in many of the competitions, and also for predicting binding energy. So this is You can read more about this method by Polo Lam, Max Totroff, and Ruben Abagayan. The 2 different papers in 2017 and 2018 in this journal. Okay, so another way to perform virtual screening, docking. I had a question, actually interesting question about multiple structures in the unit cell. I'm going to get back to that at the end because that's quite useful. If you have, we'll go through that. We can separate them and put them in a stack. We'll do that at the end. But anyway, if you had a hybrid, so yeah, one way is if you have multiple receptor conformations. But in some cases, the binding pocket is not that flexible, except for maybe 1 or 2 or 3 or 4 side chains that move. Or you may have hydroxyls that you want to rotate and be flexible. So you can just use the regular grid, but then you just select residues or water molecules that you want to be flexible, and then they won't be incorporated in the grid, they will be completely explicit. So this method, so for example here we have some hydroxyls that are going to be flexible. So it's nearly as efficient as pure grid docking, and when it's used carefully it's very useful. It's very efficient and very useful for finding new leads. By carefully I mean there's no point selecting all the residues in the pocket. Not all of them are going to be flexible. You're going to be just wasting time in your screening, and you also may generate artifacts in your results. So it's best to, if you're in a fortunate position and you have a multiple conformations and you can see that one residue is particularly flexible, then you can just select that one or 2, and that will be fine, and you can use it that way. As an example, we have the fibroblast growth factor here, and we're going to look at this example. So in this example, if you read in the APO structure, the green one, this lysine residue that comes off this glycine-rich loop, basically if you took that pocket and tried to dock the inhibitors, there's no way that you could dock them, because in order for this ligand to dock, this lysine has to move out very slightly to do that, for the ligand to be able to dock correctly. So if you were just in the situation with the APO structure, there's no way you'd understand how the binding mode of these inhibitors work. So I'll show you how to do this in ICM. So we have this fibroblast growth factor receptor kinase, and here's the lysine in question in the APO structure. This is the APO structure. So I just want to show you, and we have a number of inhibitors here. So if you dock just regular docking, then you dock them and there's no way that these inhibitors are going to be able to bind correctly. So they get blocked by the lysine basically, and the scores also represent are pretty low for this structure, so -17, -12. So you need to allow some flexibility of this lysine. How do we do that? So to do that, it's pretty straightforward. One good thing would be to find the pocket, so go to Tools, 3D Predict, ICM Pocket Finder. Okay, and this is the ATP pocket. So you can see it's quite, I mean, if you were just visually looking at it, you would probably question whether the pocket is a reasonable size to dock these inhibitors. These are quite large. So you may think about opening up. Fortunately, we do actually have holo structures, so we know that in this case that lysine does move. But to make it flexible during docking, we set up docking again. We go to Docking, New Project, and we can say flex sidechain for the project name. The receptor object is 1FGK, and then we'll define a site around the G_pocket 2, this red one. Just need to find it in the drop down list. You can see the green crosses. Go OK. Go. The box is OK for this example. Now it's making the maps. I shouldn't have made the maps because you have to remake them because we're going to define that lysine as flexible. So anyway, the maps are made. That's pretty quick. So we would like to tell ICM that we would like to keep this one single. Let's just zoom into that lysine. This one lysine residue we would like to keep flexible during docking. So we would select that residue. Click here, this residue selection button. It's a green cross. Just click on this residue. And you can see that all the atoms within the chain, in that side chain, have been selected. So then we go to docking menu, and we're going to do flexible docking. And rather than 4D grids, which we did earlier, we would do we'll use explicit groups. Click on explicit group, and just one second. Yeah, just click on that one residue. So you have one. I had a different selection made as well. So this is one single residue selected. We're not choosing hydroxys and waters. We want to have the full side chain to be flexible. So we go OK. And in the terminal window, you'll see a message to recalculate the maps. So it will recalculate the maps and exclude this lysine region because this lysine is going to be explicit in the docking. So then we need to go docking and remake receptor maps. Just go OK, and that will remake them. And then you would go docking. You can dock a chemical table, or you go docking. Well, in this example, we have all the inhibitors as a chemical table, so it's convenient. Or you go docking, set up batch from VLS from an SDF file, and split. If you're doing a large-scale virtual screening, you would choose there. And because it's we would also need to increase that thoroughness value to 10 or something in that range. So you go docking, and the docking runs, and you will then see the results. I just have the results already made. So if you go File, Open, Flex Residues. So in the same way, we get the hit list. You can rank it by score, and then you can toggle through each one, and then we can see, hopefully we'll see that lysine residue shift. You can see it move depending on which ligand is bound. So okay. So that's the hybrid docking. So if you were with us at the beginning of this series, we were talking about the 3D ligand editor. So that's an environment which is kind of after screening. So you'd have a lead compound in a crystal structure. You may want to optimize it for specificity, or you're trying to optimize the affinity of it, and you can make modifications in 2D or 3D and immediately see the effect of those modifications on ligand binding. And so what we didn't show 3 or 4 weeks ago was how to do the flexible docking in the ligand editor. So I'm just going to quickly show you how to do that. It's useful also for specificity. So you you could maybe read in different isoforms of your receptor and then design specificity based on multiple conformations. So to do that, we would just go, it's pretty straightforward in, just going to open it. So we can do both the 4D docking and the explicit docking So in the ligand editor, so for the ligand editor, we use this lig edit tab, and we may remember that we basically work our way along the buttons. So the first thing is to tell ICM where the pocket is, or the ligand that we want to optimize. So you can click here. This is the estrogen-related receptor structures again that we used earlier. I'm just going to choose one of the ligands. It doesn't matter which one. But then the key thing is the next button along, which is this is for 4D docking in the ligand editor. So we're going to use multiple conformation receptors. So we need to tell ICM which receptors we want to use. It's going to select the A chain of each of these structures, and then go to Setup Receptor. And you can go back to the videos if you want to read more, hear more about each of these options. But the receptor object, we're just telling them ICM that the 3 structures are going to be our receptor, and then we make a box around the existing ligand that we chose in the pocket, and go OK. So you can see in the terminal window that it's making maps for each of the structures, docking energy maps. Okay, and then you get something like this. So this is basically, I was going to change it to pretty view, but sorry. So basically in the ligand editor now we have multiple conformations. We have this little tab where you can toggle through each conformation, and you can see the docking score for each one. So this ligand is seriously not happy in this structure. It's got a very high VLS score. So if we toggle through, we probably hopefully find which one. You have to reevaluate the score here by clicking here. So this is probably the structure that this is actually the agonist, ERR agonist, and this is the agonist structure. So if you want to go to the inverse agonist structures, we can toggle through each one. So now you can use the editor in the same way as I showed a few weeks ago. You can edit the ligand in, let's just undisplay that. So you can make modifications to the ligand using this panel. So you can add a bromine here, and then you can assess that score, that ligand within each of the structures. So you can delete atoms. We showed all this the other day. Delete atoms here. You can do a virtual screening of multiple substituents just by selecting them here and just clicking on the point, and it will dock them. But if you want to dock this ligand to all 4 structures, you go to the docking menu, docking button here, click and hold, and choose redock in multiple receptor conformations, 4D. And then that will dock a similar way. You'll get a table with a similar thing as we saw before, where it will tell you which receptor conformation it prefers, 4D. So to do explicit docking in the ligand editor, that's going to get a new session, ICM session. Basically, we can just do the hybrid example again with that lysine. So to make that lysine flexible, we simply we save so go to lig edit. We can use one of these ligands as a starting point, and then the receptor is, we're only using one receptor, the one FGK, and this may make box. So this is making the box for the energy maps, and then we can sorry, just gonna then to make that light, you can see actually this is the inhibitor from the holo form. You can see that it makes a big clash with this lysine here. So we can make that lysine flexible during docking. as we get a similar way, we select that lysine and then go to Lig Edit Advanced in the Lig Edit tab and choose Define Flexible Receptor Groups. You can choose more than one if you wanted to, but just be careful, and choose full chain. It's going to make that one residue flexible, and so it will dock it in the same way. So now when you do any modifications in the Modification tab and then redock, that lysine will always be flexible. Okay, so that's in the ligand editor. So far we've been in the situation whereby we've had crystal structures with multiple receptor conformations, or we've generated multiple receptor conformations from MD or some other method or molecular modeling. But sometimes you're in a situation where you don't have that luxury, and you may sort of be kind of aware that your pocket is maybe slightly collapsed a little bit. It may be an APO structure similar to the one we saw with the fibroblast growth factor receptor. So inbuilt into ICM is a method for basically just opening up your pocket and building an ensemble of structures. And that method is called SCARE. It should actually be read SC for scan, alanine, refine, SCARE. So the basic observation, this is described by Giovanni Botticoni and Max Totroff and Ruben Abagai in his 2008 paper. I can provide that. This is all embedded into the graphical user interface. So basically the observation is that in a lot of cases, steric clashes caused by induced fits generally involve maybe one or two side chains within the pocket. So this method basically goes around the pocket and mutates residues to alanines and opens up certain regions of the pocket which would otherwise be inside, inside the pocket. So you basically use it, it opens up, you get an ensemble of structure, of different variants of the pocket, different shapes, and then you take a known ligand and then dock it to the pocket, and then the top scoring post from each ligand is refined, and then the pocket is refined and scored, and then you find a decent pocket. It takes approximately 2 hours on a regular desktop to run this. So this basically just opens up the pocket. So one example we have online is this structure, which I think is a J&K kinase, and basically there's an inhibitor that should be, so this is one conformation of the structure, and you can see these two phenylalanines, they inside the pocket, so there's no way that this inhibitor could actually bind, because it blocks this lipophilic channel that it uses actually, and this is the crystal structure with it bound. It blocks this channel where it comes out here. This is the lipophilic region. So it'd be nice to, so if you run SCARE and mutate these two into alanine, the pocket shape is modified enough with this extra region in order to dock a near-native, so you can redock this ligand and it will dock in a near-native way to the crystal structure. As I said, it takes 2 hours to run, but practically speaking, to do that, all you have to do is set up your docking project and then go to Flexible Receptor and choose the SCARE setup and then run it. If you go to our website, there's a detailed tutorial on how to run that. So that's just one example. And so inbuilt into ICM, if you don't have a MD simulation structure or crystal structure or NMR structure or other multiple conformations, there are some methods inbuilt into ICM to generate new an ensemble of structures. So one way would be to model side chain conformations. You could build homology models. You can also build loops. And if you go to our webinar archive, there's a webinar on each of these topics. Another approach is to take the binding pocket, place the ligand inside it, and then model the pocket around the ligand. It's called ligand-guided modeling, and it's been used in ICM for quite a few years now, but in the new release coming out soon, 3.8-7C, there is a graphical user interface option to do that. So you basically provide ICM with a table of known binders and maybe non-binders, and then you can use those binders to model that pocket. Another way would be to generate pocket conformers with normal modes. And so in the fall, we're going to have a series of webinars focusing on these things, on molecular modeling, and you can look at some old webinars if you want to interested in that, or go to our documentation. You can see that. So, for example, with normal modes, you will. That's gonna. So, just an example with normal modes, you'd get a stack similar. You can run it, go to mole mechanics, and then generate normal mode permutations, and you would get a stack like this. You can see the flexibility, and you can use that stack for 4D docking as well. This is aldose reductase, and there's this loop region here that comes in and out of the pocket if you look at multiple conformations of this protein. So you can also run loop modeling. So you could select this this region, this loop region, go to mole mechanics, choose loop, and so just going to make this so select the loop region. So green crosses around the red loop we have here. Go to mole mechanics, go to loop modeling and sampling. So there's two options. There's interactive, which will go to the PDB and find loops with similar size to this one and graph them, and it'll graph that loop on. Or you can do high precision batch, which will do a full global optimization of the backbone and the side chain, and you get a stack of conformations. So then you can choose, in this case, you could choose 3 or 4 or 5 and then run the 4D docking approach. Okay, so just an example, if you want to read an interesting review article, this is by Katrich, he's at USC, and Ruben Abagayan, and it basically describes an interesting story where they used ligand-guided modeling to predict the agonist conformation of a GPCR, and then a couple of years later that model was shown to be correct when the crystal structure of the agonist, I think it was the A2A adrenergic receptor GPCR. So that's a good example. These papers describe using normal modes for generating ensembles. Okay, so just want to thank you very much for your support of the webinars, and please, I'll try to get back to, I've got quite a few questions on different things. Some of them definitely have to get back by email, but others I'll try to answer in a little bit. The next webinar is next week, actually, covalent docking, and we'll show some examples of how to do covalent docking next week. Okay, so thank you very much, and I'll try and answer some questions.